Hello, I'm Rustin and I'd like to talk to you about the project I worked on at NIH 2 summers ago, entitled Functional Near-Infrared Spectroscopy for Real-Time Neurofeedback. I worked on this project at the National Institute of Child Health and Human Development with Drs. Frank Amiot, Laleh Najafizadeh, and Dr. Amir Gençbakhsh. The goal of my project was to create a real-time neurofeedback loop to combine the FNIR system with brain-computer interface. The FNIR system is a device that uses near-infrared light to image parts of the body, which in this case would be imaging the brain. FNIR stands for Functional Near-Infrared Spectroscopy. So I'd like to use this neurofeedback loop to help treat patients with autism or traumatic brain injuries or other kinds of problems with the brain, to help train certain parts of their brain to work more successfully. So FNIRS works based on the principle that near-infrared light passes easily through tissue, has a very low absorption rate, and the thing is, oxygenated blood and deoxygenated blood have different absorption coefficients. So when we send light through, we can read the amount of light that bounces back to the detectors and see how dense the blood that the light went through was. And by knowing that, we can know whether or not that part of the brain was being activated at the time that we were looking at it. We used the modified Beer-Lambert's law to find how much of the blood was oxygenated and how much was deoxygenated, so that we could pick up on these activations. At NIH, I used Tekin CW6 system, which gives off light at 690 and 830 nanometers, and the sensor we used consisted of 4 sources in the center sandwiched between 8 detectors on the outside, and this would be able to detect activations in a fairly large region of the motor cortex, which is what we were looking at in our experiment. We conducted a simple finger tapping experiment in which we monitored the patient's brain activities during a simple finger tapping. So we were able to look at activations of the motor cortex during this time. After learning MATLAB from my colleagues at NIH and Drexel University, I was able to write an activation finding algorithm that could detect activations of the brain in real time. So this algorithm has a delay of about 5 seconds between the stimuli and the detection of the activation of the brain, which is due to delay in the blood reaching the parts of the brain, as well as just computing time. After studying the brain further, I'd like to isolate certain parts of the brain that are related to different disorders, such as autism or ADHD, and I'd like to improve my algorithm to have less of a delay between the activation and the detection. And I'd also like to just experiment with other studies and maybe find other parts of the brain that could be related to diseases that we could improve the function of.